Alright, today we're going to talk about transcription. The process of transcription takes a DNA segment and makes an mRNA strand from that. The mRNA strand that is produced during transcription is then used during the process of translation, where it is used to make a protein. First, let's talk about the RNA structure. RNA differs from DNA in four ways. RNA contains the sugar ribose, not deoxyribose. RNA contains the nitrogenous base uracil instead of thymine. RNA is single-stranded instead of double-stranded, and RNA is shorter than DNA. There are three types of RNA. Messenger RNA, which is mRNA, carries instructions from a gene to make a protein. Ribosomal RNA, rRNA, is part of the structure of a ribosome. Transfer RNA, tRNA, transports amino acids to the ribosome to make a protein. In the image to the right, you have the mRNA here, the ribosomal RNA here, and transfer RNA here. Transcription is the process of transcribing a segment of DNA, called a gene, into an mRNA strand. It occurs in the nucleus of the cell, and then it exits from the nucleus to be translated in the cytoplasm on ribosomes. There are three steps of transcription. The first step is initiation. RNA polymerase binds to DNA at the promoter, a specific sequence of nucleotides on the DNA. The two DNA strands unwind and separate, and so it's binding to the promoter in the yellow here, and this is the RNA polymerase. Second step is elongation. Only one strand of DNA acts as a template. RNA polymerase adds RNA nucleotides to the growing RNA strand. So as we look to the right here, we have the RNA polymerase moving along, producing the RNA strand. The third step is termination. RNA polymerase continues to add RNA nucleotides until it reaches a termination signal on the DNA. The new RNA strand is released and DNA reforms into a double helix. So once it reaches a termination signal, it releases the RNA polymerase and the mRNA strand also releases. Eventually the DNA will bind back into the double helix. Let's practice transcription. So how to transcribe. Use the complementary base pair rules for DNA to RNA. It is slightly different than the DNA base pair rules, where it was adenine binds to thymine, cytosine binds to guanine. If we have the DNA strand going to the RNA, if we have DNA is A, or adenine, it will pair on the RNA strand as uracil. If the DNA strand you read C, or cytosine, it will bind on RNA as guanine. If its DNA is G, or guanine, it will bind a cytosine. If it's DNA and it's thymine, it will bind to adenine. So the only difference is that in RNA you do not have the thymine. When you see an A in DNA, you do not pair it with a thymine, you pair it with a uracil. So let's practice. So this is the DNA strand. We have A, G, C, T, A, T, C, C, G, G, A, C. So first, we have A. So if we look over here, A pairs with U. Next is G on DNA. So G pairs with C. And then DNA has C, and that pairs with G, guanine. Next is T. So we're going to find T for DNA, bring it across, and you have RNA is A. So next we have A. A on DNA pairs with U for RNA. T on DNA pairs with A on RNA. C on DNA pairs with guanine, and that's another C, so that's another G. Next we have GG, so we look for DNA for G, and it pairs with C for RNA. Next we have A on DNA, and that pairs for U. And lastly, for DNA, you have cytosine or C, which pairs for guanine. So if you have any questions, please feel free to email me, and I will get back to you, or watch the lecture notes again. Have a good day.